Welcome to STEMpaths, where students unpack papers, study paths, and the people behind them. I'm Dr. Thornton Larson, and with us today we have our guest hosts. We have Jennifer and we have Anna, who are going to be interviewing, oops, I'm stumbling already, our guest Dr. Adam Boyko about his paper regarding genetics of huskies. You guys want to take it away?
Yeah, of course. So, hello everyone, and welcome to our genetics podcast. I'm Anna Sanchez, and I'm a psychology major with a minor in biology. And my goal is to become a clinical psychologist or a counseling psychologist.
And my name is Jennifer Gutierrez, and my major is also psychology with a minor biology. And I'm also looking forward, and my goal is to go into clinical psychology. So today we're speaking with Dr. Adam Boyko, and about his paper, Direct-to-Consumer DNA Testing of 6,000 Dogs Reveals 98.6 kb Duplication Associated with Blue Eyes and Heterochromia in Siberian Huskies, Genetics Behind Blue-Eyed Dogs in PLoS Genetics.
It's a really long title.
Yeah, the title is very informative in itself, yes.
Okay, so this study focuses on understanding why some dogs, especially Siberian Huskies, have blue eyes. So the researchers analyzed genetic and survey data from more than 6,000 dogs using a genome-wide association study. So through this analysis, they discovered a strong link between blue eye color and a 98.6 kilobase duplication on chromosome 18, in a gene called ALX4, which is involved in eye development.
And this duplicated region appears to influence how the ALX4 gene functions, making blue eyes more likely. The duplication is especially common in Siberian Huskies, which explains why blue eyes are so frequent in that breed. However, this study also found that some dogs with the duplication still have brown eyes, and that some blue eyes dogs do not carry the duplication, suggesting that an additional genetic factors also play a role.
Okay, so overall, the paper shows how a large-scale DNA datasets can help identify structural variants that affect visible traits in mammals, right? Is that how you say it? Mammals? Mm-hmm. Sorry.
Yeah, you're doing all right.
So thank you so much for joining us today, Dr. Boyko. Did that hit all the key points?
Yeah, I think those are the key points about the biology of what's going on here. It was kind of a fun study because it was the first time ever a dog DNA test was used to make a new discovery about a new gene involved in a trait in dogs before. We'd done that with humans because people have been doing DNA testing for a little bit longer. But it was kind of fun to have a dog data set that was built from, consumers testing their dog and telling us about their dog and then being able to make an association.
Yeah, that's great. So to begin, we want to learn a little about your path into this field. What first drew you to studying canine genetics?
So I started out as a computer science major. Well, actually, I started out as a physics major, and then I switched to computer science. And then I kind of missed the, being able to do hypothesis-driven science. And so I wound up double majoring as an undergrad in evolutionary biology and computer science. And so then I went to grad school, and I got to go out in the field. I got to catch butterflies in the tropics, and I got to bring them back and have colonies and do experiments and also run simulations. And so I did a lot of combination of biology and computer science. And by the time I was graduating, I sort of realized all the cool stuff that was at the interface of biology and computer science. It was really genomics. And so I started really hitting genomics hard. I got a postdoc at Cornell in a lab that did population genomics. And most of the big data sets at the time were human data sets. And so that's what I was working on. And then, I don't know, about 3 years into it, we started a collaboration with some other labs and generated some really cool dog data sets. And it was all purebred dogs that we generated data from. And that was fun. And we made a lot of really interesting discoveries. The gene for short legs and basset hounds, the gene for floppy ears and a whole bunch of different breeds. Wiry coats versus straight coats, all sorts of stuff. But then people were trying to figure out, okay, well, now we have all this dog genetic data, where do dogs come from? And that's sort of set off alarm bells to me because, that would be like studying European human royal families to try to figure out human origins. Like, you don't have the right genetic diversity to answer those kinds of questions. And so that, I was like, there's 1 billion dogs in the world, and most of them are not purebred dogs. They actually would be living in natural free breeding populations, and that's where you're going to see most of the genetic diversity in dogs, that you're going to be able to tell where dogs came from. And so I was fortunate enough that Cornell was willing to fund a pilot project. My brother and I worked on it together. And we basically traveled around, him or I or us together, and just collected samples from dogs in Africa, in the Middle East, in East Asia, in Central Asia, in Europe. We went to Peru, not that we think that dogs came there, but we kind of wanted to know what happened to the Native American dogs. We did some island hopping in the Pacific to try to figure out if there were still indigenous, Polynesian and Melanesian dog populations left on any of the islands. And so, that was a really fun project. It was, we were kind of the only ones that had those sorts of samples and could do that sort of analysis. And then a lot of people caught wind of the project and were like, hey, can my dog be part of your study? And can you tell me what breeds are my dog? And we're like, well, I mean, we can. We've got the DNA, but, we don't really have a customer support infrastructure for dealing with all of this and building a website and things like that. And so, so we kind of changed gears. It took a little while of talking to Cornell and talking to other companies, and nobody really wanted to do what we wanted to do with dog DNA testing. And so we wound up starting a company called Embark. And we started in 2015. We started doing genetic testing in 2016. And then in 2017, we saw this association. This was the first thing that popped up. Nobody had ever done an eye color study in dogs before, but it was easy for owners to tell us what color their dog's eyes were. So that was one of the things they told us among the litany of things. And so then we wrote it up, and we got this PLoS Genetics paper, and now people can test for eye color with Embark or with even other genetic tests, since we published how to do it.
Okay, that's nice. So you guys have other tests other than eye color, or was eye color specifically what you wanted to focus?
No, we had no idea that we would make a discovery in eye color starting out. And so when we started out, we tested for a lot of the known coat colors, as well as known disease markers. So there's hundreds of genetic diseases in dogs that we know the DNA variant that causes that disease. But a lot of them are recessive, so you don't know if your dog is if you're going to breed a dog, you don't want to breed a carrier or do another carrier accidentally. So the testing's important there. But also, if you have a pet dog, you want to know if your dog is going to develop one of these disorders later on, even if you're not breeding the dog. And a lot of times you can feed the dog a different diet or do different kinds of training or things. But at the very least, you know what to look out for. Mm-hmm. And most people just wanted to know what breeds were in their dogs. And so, we had at the Cornell Biobank a really big database of purebred dog DNA, and we had all the village dogs in there, so we could say, which breeds are in the dog, where did the dog come from, all of that kind of stuff. So it was the first time a dog DNA test was offered on a research grade array. So we could combine the research data we generated at Cornell with the customer data, because now we were looking at hundreds of thousands of markers in the dogs instead of just dozens of markers, or at most 300 markers. This is what was on the market at the time.
That's really interesting, honestly. It's always helpful to understand what motivates a research direction before looking at the details.
Yeah. And in fact, it wasn't eye color that we were interested in for doing this. We wanted really big data sets so we could start to answer questions about aging and longevity, cancer predispositions, behavioral differences. All of those things require really big data sets because there's lots of genes and genetic variants that are involved, and there's a lot of environmental noise that you have to be able to average out. Whereas if we wanted to do a study on eye color academically, we wouldn't have needed a huge data set to do it, but we still would have needed somebody to fund it, and nobody's really funding eye color work in dogs because it's not really a model for human medical disease.
Okay, sounds good.
How much interest is there actually in with the people? Obviously, you started a whole company based on people being interested in the genetic history of their dogs. and that in part helps with further discovery of these new traits down the line, correct?
Right. Yep. And some of them have been disease traits. So we identified and published, recessive deafness, early onset deafness, and radiation ridgebacks. And so now the breeders know which dogs are carriers, and there's no more deaf radiation ridgebacks that are being created because you never cross a carrier with another carrier now because you could test for it.
That's so cool.
Yeah.
Okay, so for listeners in this paper who may not be familiar with this concept, like how would you describe what a duplication is?
So a duplication is a section of a genome where the part of a chromosome got copied and reinserted into the genome. Right, so we splice out stuff in the genome all the time. There's different elements that do things. We don't want to reinsert it in the genome. We want to keep the genome as conserved as possible. But it happens, and in this case, it happened as a tandem duplication. So we have the normal part of the DNA, and then the duplicated part is right next to it. Okay. Yeah, and so everybody's got two genomes. Dogs have two genomes, humans have two genomes, all the diploids have two genomes. What do you got from mom? What do you got from dad? And so each one of those genomes can have either one or two copies of this duplication. There's other duplications of the genome where you could have more than two copies, and that happens in dogs and humans, and it can be associated with various phenotypes as well.
Okay, so one of the major findings in this work was this duplication, but how does this duplication specifically relate to eye color? What is it about the location near the ALX4 gene that makes it so significant?
Right, that's a fantastic question, because this is not a duplication that contains the ALX gene in itself. It's upstream from it. But ALX is a gene that we know is involved in eye development and pigmentation. So it makes it a really good candidate gene. So something about this duplication is controlling the regulation of that gene, like when it's getting turned on or off, or where it's getting turned on and off, or how much of it is getting turned on and off. It's really hard to work out all of that without building a transgenic organism. A lot of times people will splice something into a mouse. Now that we have CRISPR, we didn't have CRISPR when we did the study initially. So these days you would try to take a CRISPR mouse, and it's a little difficult because it's not a single base pair change. You're trying to do like almost 100 kilobases. So it's kind of tough to do that. And it would be really important to do, I think, if we saw that this was like a cancer syndrome and we didn't understand how the genes work, because like there's really a lot of interest in understanding cancer. Because building transgenics is expensive, it's easier now, but it's just not the highest priority to try to figure out all of the ways that this eye color pathway works, because there's a lot of like really interesting stuff with aging and longevity and cancer and brain development, behavior kinds of stuff where maybe it makes more sense to go further down the line. So sometimes you get to a point in a study, it's like, okay, I can offer a genetic test now. Breeders that are interested in knowing eye color or whether a dog carries something, because this is not fully penetrant. There are occasionally dogs that have the duplication and have brown eyes, but if you breed them, their offspring will predominantly have blue eyes, the ones that inherit it. So it would be good to know if this is a dog that's likely to throw blue eyes, if that was a phenotype that you were interested in. So we can test for it accurately. We really need to understand all of the mechanisms under it. In this case, we kind of decided, no, we want to find more cool stuff, and we've done that. We've published more papers about cool associations with other phenotypes and genes.
Okay. So also your paper shows that this duplication accounts for about 75% of blue-eyed dogs. So for the remaining cases where dogs have blue eyes but didn't carry the duplication, what are some possible explanations?
So it could be a blue-eyed dog for due to a different genetic variant that we know about. So dogs that are Merle often have blue eyes or heterochromia. Merle is another dominant mutation, but Merle also affects the coat color. So you get that mottled coat look, and it's not good to cross two Merles because a double Merle, it's a very light coat color, very prone to deafness and eye disorders. So that's one you want to look out for. Here we don't see any association with those sorts of negative phenotypes with this locus and blue eyes. And then the mid-F is the white spotting locus, and that can sometimes lead to blue eyes, but not as often as this locus, which really seems to be focused on eyes, and probably because it's this ALX gene, which we know is important in eye development. But we also have other dogs that have blue eyes, and so there must be other genetic variants, or that, I don't think there's an environmental thing that's causing the blue eyes, but you can't, I guess, rule that out without really studying it. So we found the most common one. Now that we have a bigger data set, we could probably go back through and see, are there any others maybe that pop out? That'd be an interesting follow-up study.
And does that also explain kind of like in the opposite situation where dogs did carry the duplication, but they still had brown eyes?
Well, again, so I think what's happening is when in an embryo, you have cells that are migrating during development, right? And particularly for these pigmentation, for the cells that are the progenitors of the pigmentation cells, if they migrate more slowly, then they don't get to certain parts. So usually that's going to be the tip of the nose, or the very front of the chest, or the tips of the paws, or the tips of the tail. And so that's why you see white there in dog. Like, that's why in dogs where they've slowed down that migration of those pigmentation cells, that's where the white starts out. And then, of course, dogs get more white and slow down even more. And so I think that you have some sort of interaction with that sort of development thing, and it didn't wipe out the brown eyes in some of the dogs just by chance, because development, lots of things can affect that. So this isn't like when you cross a black lab, with another black lab, and if you know the genotypes, you could predict, are all the offspring going to be black, or are some of them going to be yellow, or are some of them going to be chocolate? You can have all those different combinations, and it's like 100% penetrant. Like, what it's going to be.
Okay. Well, thank you so much for your answer. And that honestly helps you clarify how complex phenotype expressions can be. Sorry.
Great.
So looking into your methods, your team used a combination of approaches, including microarray intensity, read depth analysis, and PCR. Could you walk us through what each method kind of contributed to confirming in the duplication?
Okay. Yeah, so a microarray, I don't have one right within reach here. I could show you. It's a little tiny array, and you can run DNA over it, and it's got little beads that are supposed to match up to specific parts of the genome. And so we chose, 200,000 parts of the genome that we knew were variable. And some of them we knew were associated with stuff, but most of them we use it for ancestry inference, but we don't have any expectation that it's going to be associated with anything. And from that, you can tell which variant the dog carries in each of those 2,000 or 200,000 places in the genome. Now, of course, the genome for a dog is 2.3 gigabases. And so it's got lots of variants. It's got tens of millions of places where there's letters that are different between different dogs. So we're only sampling a small part of them. But because there's only 39 dog chromosomes, like, we've got thousands of markers on each chromosome, and we sort of spaced them out so like the whole chromosome is more or less evenly covered. And so in this case, we can't look for a duplication using the array, but that duplication is on chromosome 18, and it happens to be next to 4 markers. And so you almost always inherit those markers and that duplication together. And so even though we knew nothing about, what's the causal gene for eye color here for blue eyes, and we didn't have the variant that causes the blue duplication on the array, we still got this association because other markers in that region are linked to it. And so they're going to associate too. And so then once we got that associated, that's like getting the zip code. Okay, we know now the mutation has to be in this zip code. Okay. And so we looked at sequence data then to see, are there any mutations on this haplotype that break a gene? No, we don't see anything like that. Are there any like big deletions? No. Is there a duplication? Oh yeah, there's this really interesting duplication. It's got lots of regulatory stuff in it. And then we could do this experiment where we can ask the owners who didn't tell us about the dog's eye color. So a validation experiment. Mm-hmm. Hey, your dog would be interesting for this study. Can you tell us which color eyes your dog has? And sure enough, when that came back, the duplication was even more associated with eye color than any one of those variants that we had initially. So that gave us a really good idea that it was that duplication.
Okay. So zooming out of it, like what can discoveries like this one about a specific trait such as eye color teach us about broader concepts in mammalian genetics?
So it tells us that a lot of the variants that underlie phenotypic diversity aren't ones that change the protein. They're ones that just change the regulation of the gene that codes for the protein. And so that makes sometimes identifying causal variants difficult or proving it. But it makes sense that, a lot of times most of the time it's better for evolution to make small changes in the amount of a gene that's working that you have than to make big changes in the gene itself that might break it or might cause it to have really, not working effects. And so we see this for a lot of dog phenotypes, a lot of the body size genes that we know about. We know they're changes in the regulation of the gene that are leading to the differences in body size, not to the protein itself. There are exceptions. But a lot of the disease alleles are usually breaking a gene. And so we see a lot of recessive disease alleles, and those are in the gene themselves a lot of times.
Hey, and at the time, your study was the largest canine GWAS. How do you see large-scale data sets influencing future research in either dogs specifically or in other mammals?
I mean, they've already been a huge boon for human genetics. So we now have had GWAS studies in humans with millions of data points. And that gives us a really good idea of the genes that underlie, differences in height or cardiovascular risk. When we have these really big GWAS, we get, for humans, lots of genes that show associations. And even when we can't identify specifically what the mutation is that's causing that association, it gives us an idea that this gene must somehow be involved in this disease process. And so even if we don't know the actual mutation and we don't know, and it doesn't really let us predict who's going to get the disease or not because it only influences it by like 1%, we do know that gene has to have something to do with the disease. And it gives drug companies something to look at, or researchers, whether they're academic institutions or pharmaceutical companies. And so we've gotten a lot of new drugs from genomic data that showed us that there was an association, or other kinds of therapies, gene therapies or things like that. But you have to have those big data sets before all of those associations start to make themselves known. And it'll be really exciting as we're starting to go down the path of doing that in dogs, to know why do some breeds of dogs get different cancers and higher probabilities of cancer than other breeds of dogs. They're all more or less being raised in the same household environment. So it has to be a genetic, reason. And we know that big dogs age faster than small dogs do, but we don't really know the genetic mechanisms by which that happens and whether there's any other sorts of genetic factors independent of size that are influencing aging in dogs.
Okay.
Interesting. I know they've got a lot more questions, but just to respect your time a little bit, I think we need to move into the 3 wrap-up questions that we ask every one of our guests, just to make sure we're sticking to a good time schedule here. So if you guys want to ask those last 3.
So first, what is one piece of advice you'd want to give your undergrad self?
I mean, there's so much advice you want to give yourself about, where to invest it and all that kind of stuff.
Right. What would you listen to at that time too, right?
What I listened to, I don't think I would have listened to anything. No, but as an undergrad, I knew I wanted to do something that would leave options for me to pursue different paths, because I knew I didn't know exactly what I wanted to do. And so that's kind of why computer science appealed to me at first, and then being in the field and being able to do scientific inquiry and hypothesis testing that way, I didn't know if that was just going to be a side gig and I just, want to be a software engineer for the rest of my life, or whether that side gig would turn into something else. And as I got to my senior year as an undergrad, I decided I wanted to be a scientist that was good at computers rather than being a computer scientist that had a hobby of being a scientist. And so that's why I did the PhD in biology.
Okay. So if you weren't doing this, what would you think you'd be doing?
I'm sorry, I missed that.
Oh, sorry. If you weren't doing this, what would you think you'd be doing, like, right now?
I mean, I think I would probably have been a software engineer, and I probably would be burned out by now. Now I've worked with some pretty high-level software engineers from about my same time.
All right, so last question. What is one thing that you wish more people understood about scientists or science?
Great. I think people see the neat story at the end, and then a lot of times, like for instance, for this study, we got a lot of criticism. Why is taxpayer money going to fund why some Siberian Huskies have blue eyes? And the fact that people can be so anti-science and anti-funding science just as a knee-jerk is just shocking to me, because this study literally was a startup company, got customers to pay to have their dog's DNA analyzed, and then the company paid the scientists in the company to see if there was any cool genetic associations, and then published this result. And so instead of the taxpayers paying any money towards it at all, it was free research that then got shared scientifically, but it still got criticized because people were like, "There's so much stuff you could do studies on," and of course, we didn't go out to do a study on iColor. We asked owners lots of different stuff, and this was just something that was easy for the owners to phenotype, and happened to have this really strong association that was, right at this genetic variant that made a really good, compelling case that it's likely to be the causal variant. So that's why we published this one.
All right.
That's such a cool journey, I think, just the idea of you collecting all this data through both a combination of data collection of the tissue and the genes mixed with surveying the pet owners and just seeing what's there, and then you catch a low-lying fruit here or there that proves the essentially proves the pudding or whatever it is, but the proof in the pudding or whatever it is.
Right. I mean, yeah. So, and when I was a postdoc and we did the canine mapping study with the different purebreds, we took 50 different measurements from all of the different breeds, and then we just saw which parts of the genome correlated with different measurements, and that's how we got short legs, short snout, big body size. But then when we looked at the DNA, we're like, well, there's this other region that really seems to be strongly differentiated across the breeds, but it doesn't match up with any of the measurements that we have, right?
Right.
And so I had to do a Google image search of the different breeds that had one version or the other version, and it was the gene for floppy ears. So some breeds have floppy ears, and then some breeds have the ancestral wolf prick ears. And we didn't go out to do a floppy-eared study, but that happens to be the low-hanging fruit in the genome. That's the biggest signature of selection in the dog genome, because breeders have to fix the ear conformation within each breed.
That's so crazy. Well, I want to thank you, Dr. Boyko, for making the time today. I know you're incredibly busy. You have a really cool company, a really cool gig as far as I'm concerned. I think the students, I want to thank Jennifer and Anna also for putting together the questions, finding your paper to go over.
Questions. I love talking about dog genetics, so thanks so much.
So thanks again. This has been STEMpaths. I'm Dr. Thornton Larson. Thank you, and we'll see you again next time.
Thank you.